Okay, so for question 5a, describe two classes of antibiotics that inhibit cell wall synthesis. Our two main classes are going to be beta-lactams and glycopeptides. Glycopeptides, namely vancomycin, teicoplanin, and telebenzin. Beta-lactams, on the other hand, can be further subdivided into four classes, mainly penicillins, monobactams, cephalosporins, and carbapenems. Penicillins can be further subdivided into aminopenicillins, anti-staphylococcal penicillins, anti-pseudomonal penicillins, and standard penicillins, which we are all familiar with. As for the next question, compare the mechanism of action of an example from each class. Okay, the examples we should be going with is going to be penicillin, go with vancomycin. Those are the examples given in the lecture notes. Before we understand the mode of action of penicillin and vancomycin, let us have a look at cell wall synthesis. Well, cell wall synthesis is going to be initiated through two precursor molecules, namely NAM and NAG. What is NAM? NAM is sugar, namely N-acetylmuraminic acid, and NAG is N-acetylglucosamine. NAMs are attached to NAGs via 1,4 beta-glycosidic bonds. Okay, beta-glycosidic bonds. Attached to each of these NAMs and NAGs, I haven't drawn them out, all of them out here, but you can see attached to this NAM we have a pentapeptide chain. Attached to this NAG we have a pentapeptide chain. So the question is, how are we going to link layers of NAMs and NAGs together? Well, this is going to be through cross-linking or transpeptide bond formation between molecules of D-alanine and molecules of lysine. As we can see, we have two molecules of D-alanine down here, molecule of lysine right here. To achieve this, we use this transpeptidase enzyme, which is going to catalyze bond formation from one molecule of D-alanine, one molecule of lysine. During this bond formation, one molecule of D-alanine here is going to be cleaved off. As for the mechanism of action of penicillins now, penicillins basically work on the premise of directly inhibiting transpeptidase enzymes. So it binds to the active site of transpeptidase enzyme, and why it can bind to the active site is because the molecular structure of penicillin and the molecular structure of D-alanine is very similar, so similar that they can be superimposed onto each other. Hence, penicillins will directly bind to transpeptide enzyme, preventing it from working on these pentapeptide chains. As for glycopeptides are going to inhibit D-alanine directly by binding to D-alanine. Transpeptidase enzyme is not going to be able to work with D-alanine to create these transpeptide bonds. Now for the mechanisms of resistance. Well, for beta-lactams, the mechanism of resistance is going to be via beta-lactamase. Beta-lactamase is going to work to break down the beta-lactam ring in beta-lactams like penicillin and cephalosporin. We also have mica genes, but this is only found in methicillin-resistant bacteria. Methicillin-resistant bacteria have mica genes which code for a variant penicillin binding protein or variant transpeptidase enzyme, mainly penicillin binding protein 2A. It's called penicillin binding protein 2A because of its increased size or increased molecular weight. As for glycopeptides resistance mechanisms are based on Wan genes, and Wan genes are genes that are activated in bacterial cells when they come into contact with glycopeptides. The first Wan genes to be activated are Wan-R and Wan-S. Wan-R and Wan-S will then further go ahead to activate Wan-H, Wan-A or Wan-B, and Wan-X. Wan-H functions to convert pyruvate to lactate. So what is the function of this? Well, converting pyruvate to lactate creates an alternative ligase. As for Wan A or B functions to convert D-alanine or rather synthesize D-allylactase instead of D-allylalanine. And as I said before, glycopeptides work on the premise of binding to D-allyalanine. Hence, formation of D-allylactate will prevent the glycopeptides from working on D-allyalanine or the pentapeptide chain. As for WAN-X is going to function to break down the D-allyalanine proteins, or rather hydrolyze D-allyalanine, preventing glycopeptides from working on D-allyalanine.